This first lesson is going to focus solely on the anatomy of blood vessels by comparing arteries, veins, and capillaries, and then understanding the flow of blood through these major types of blood vessels. So, as a brief overview of what we're going to cover, we're going to look at the anatomy and function of blood vessels based on the type of blood that they're going to be responsible for transporting. We'll look at the flow of blood through the cardiovascular system and some of the challenges that are presented by trying to distribute blood to every cell and tissue in the body. We'll look at the structure of an anastomosis, as well as capillary beds and the types of capillaries that are found throughout the body. Now, to begin with, I'm going to start by using this diagram that took me hours to draw, and we're basically going to use this to understand the network of blood vessels that extend from the heart and go to all of the different cells and tissues in our body. So what you see here at the center is basically the right and left heart, with blood vessels colored in red, representing those that carry oxygen-rich blood, and blood vessels that are colored in blue that carry oxygen-poor blood. Down here at the bottom of this diagram, we have an organ. It could be, I guess I drew it shaped like a liver, but it could be any organ or any tissue in the body because remember, every cell and tissue in our body needs a blood supply to survive. So to start out with, we have coming directly off the top of the left heart, the aorta. The aorta is going to extend as we go further and further away from the heart into the distal parts of our body. This will go into smaller blood vessels called arteries, and then arteries will gradually turn into smaller arteries called arterioles. Now, these arterioles are going to be responsible for diverting blood to specific organs. So as we get into this organ that you see here, we get into a totally different type of blood vessel called capillaries. Now, capillaries, you can see an important thing happening here. We see the blood supply turning from oxygen-rich to oxygen-poor. So there's going to be an important thing that occurs here that happens only in blood capillaries and nowhere else. And then coming out of this capillary bed, we're going to have venules, which progressively lead into larger blood vessels called veins. And then eventually we work our way back to the top of or the bottom of the right heart to return that oxygen-poor blood back to the heart. Another interesting thing that you should take away from this diagram is how blood pressure changes as you go away from the heart towards cells and tissues. So if you were to track the blood pressure as it leaves the left heart and goes towards this organ that you see here, the highest blood pressure in the human body is seen here in the aorta, and that's usually around 95 millimeters mercury. That's considerably higher than the typical blood pressure that you measure in your arm. So as you go into smaller arteries away from the aorta, the blood pressure is around 80 millimeters mercury, and that's the typical blood pressure reading that you would receive if you were using a sphygmometer on someone's arm. Now, as you go further away from the heart down into progressively smaller arteries called arterioles, the blood pressure drops even further to about 35 millimeters mercury. And by the time it crosses through this capillary bed to the other side of this organ, the blood pressure is about 15 millimeters mercury in venules. And by the time it goes back to the right heart, the blood pressure is so low that it's almost non-existent. So by the time it reaches the right heart, it's about 5 millimeters mercury. So almost no blood pressure at all. So one of the main concepts I'm going to try and drive home to you from this lesson is, in the absence of blood pressure, how do we return blood to the heart from cells and tissues? So that's one major emphasis to take from this lesson. And the other is going to be related to the whole point of transporting blood through our bodies anyway. So hopefully it makes sense that we are transporting blood through all of this extensive network of blood vessels because we want to deliver oxygen to cells and tissues and remove CO2 from cells and tissues. And as a result of that, the only place in the cardiovascular system where you can exchange oxygen and CO2 between cells and tissues is in capillaries. That's the only type of blood vessel designed for the exchange of these materials. Everywhere else, regardless of whether it's an artery or a vein, cannot do that. Anatomically, they are not capable of allowing that basic function. So we rely on the use of these capillaries for that exchange, which is very critical for survival of cells. Now let's summarize what we've gone over so far. We have 5 major categories of blood vessels, including the arteries and arterioles, whose main job is to transport oxygen-rich blood, usually, from the heart towards cells and tissues. In other words, away from the heart. We have capillaries whose sole job is to allow for the exchange of oxygen and CO2 between cells and tissues. Whereas venules and veins are going to be primarily relied upon for transport of oxygen-poor blood away from cells and tissues back to the heart. This little diagram that you see here on the right basically shows you how blood pressure changes as we go from the aorta down into smaller arteries, arterioles, and then through capillaries. And then coming out through the other side of the capillary bed, you see that the blood pressure continues to drop until it almost reaches 0 by the time it comes back to the heart. And that was basically described to you in that drawing, or at least my attempt at a drawing, of the cardiovascular system on the first slide. So now we're going to begin looking in more detail at the anatomy of blood vessels by exploring the tunics or layers of blood vessel walls. So there are 3 distinct tunics that comprise arteries, veins, and capillaries. Always going from superficial to deep, we have the tunica externa, followed by the tunica media and the tunica interna. Each one of these tunics or layers is going to be comprised of different types of tissue. The tunica externa consists of combinations of 2 types of tissues, including dense irregular connective tissue and elastic connective tissue. In some areas of the body, the tunica externa is so large that it requires its own blood supply, its own set of blood vessels, to ensure that it gets adequate oxygen for survival. This is called the vasa vasorum, which literally stands for the blood vessels of blood vessels. And that's unique to this type of layer of the blood vessel wall. It is the external elastic lamina which separates the tunica externa from the tunica media. The tunica media is principally comprised of smooth muscle. This represents a very important regulatory structure found in blood vessels. Smooth muscle has the capability of obviously constricting and relaxing to help adjust the overall blood vessel diameter as well as the blood pressure that's running through that blood vessel. The relationship between the blood vessel diameter and blood pressure is inversely proportional, meaning when the blood vessel diameter increases, this is because of smooth muscle relaxing, and that allows blood pressure to decrease. Similarly, when smooth muscle constricts, this helps to narrow the diameter of the blood vessel, and as a result, blood pressure increases in that area of the blood vessel. Separating the tunica media from the tunica interna deeper to that is the internal elastic lamina, which is technically the superficial layer of the tunica interna. The tunica interna also consists of 2 additional structures, including the endothelium and basement membrane. The endothelium is principally comprised of simple squamous epithelium, which allows for the diffusion of gases more easily in certain areas of the body. To add to our knowledge of blood vessel anatomy, it's important to understand that arteries generally have thicker tunica externas, tunica medias, and tunica internas, in addition to having larger blood vessel diameters, compared to veins which have appreciably thinner tunica externas, medias, and internas. One structural detail that is unique to veins is the presence of valves. Recall that the function of a valve in the heart is to prevent the backflow of blood from an area in which the blood has already came. So then to return to this question, how do we return blood to the heart in the absence of blood pressure? One of the structural mechanisms that we can rely on to accomplish this feat is through the use of valves that are found in veins. This ensures that when we are sending or transporting blood through veins, the blood is not going to go back the same way it came and is only going to go in one direction, and that is towards the heart. Recall that capillaries are a unique type of blood vessel that allow for the exchange of gases and other substances across the blood vessel walls. It is because of this function that capillaries have a unique anatomy associated with them. In essence, they have only a tunica interna, which is comprised principally of just an endothelium and basement membrane. This facilitates the exchange of these substances across the blood vessel walls. On the following slides, we're going to begin looking at specific types of arteries and capillaries and their important structural and functional details, beginning with elastic arteries. Elastic arteries have the largest diameter of any blood vessel type in the human body, and as a result of that, they have to also withstand the highest blood pressure forces in the body as well. Elastic arteries include examples like the aorta, the pulmonary trunk and arteries, the brachiocephalic artery, the subclavian arteries, as well as the common carotid arteries that are part of the systemic circuit. The elastic arteries are unique structurally because they are comprised of thickened tunica externas and also have reinforced external and internal elastic laminas. These structural details give these blood vessels needed elasticity so that the blood vessels can basically expand to accommodate higher blood pressure forces in these specific areas of the body. The example that you see on this slide demonstrates the functionality of the aorta in relationship to the left ventricle. There are tremendous blood pressure forces that are created as the result of the left ventricle ejecting blood during ventricular systole. The aorta stretches to accommodate these enhanced blood pressure forces and momentarily stores this as mechanical energy, basically functioning as a pressure reservoir that is then released and helps propel blood further and further through the cardiovascular system. This is one of the important functional advantages of elastic arteries. The most common type of artery in the cardiovascular system are muscular or distributing arteries. These include examples like you see here on the screen, including the brachial and radial arteries. These are typically the arteries where we measure blood pressure from using blood pressure cuffs. So what is unique structurally about these types of blood vessels is that they have a significantly enlarged tunica media. Approximately 75% of the mass of the blood wall is just the tunica media in these blood vessels. Because of that, they can adjust the blood pressure significantly by just changing the diameter of the blood vessel. A cardiovascular structure that is associated with groups of arteries in many different places in the body is a structure called the anastomosis. An anastomosis is basically a union of blood vessels, and these are important because it provides alternative routes for distributing blood to the same area of the body. So an example that you see here is blood supply to the right colon. Now, if, for instance, the right colic artery becomes blocked due to a clot, becomes compressed or damaged in some way, we can send blood to the same area of the colon through the use of the middle colic artery that you see here. Similarly, a very common anastomosis, one of the most famous in the body, is the circle of Willis. So go back to your notes about the structure of the circle of Willis, and you can see the different routes for distributing blood to almost the same area of the brain. So the anastomosis is very important for alternative routes of blood supply or distributing blood to a similar region of the body. Furthest away from the heart are going to be the smallest types of arteries in the human body, which are called arterioles. Arteriole literally means little artery in Latin. Arterioles are responsible for bringing oxygen-rich blood to all of the different organs and cells and tissue beds in the human body. Using this diagram that I've created here, we're going to understand the flow of blood into capillary beds. The flow starts with arterioles, which then go into a meta-arteriole. The meta-arteriole represents the distalmost point of an arteriole that is responsible for diverting blood to a specific organ or specific set of tissues or cells in the body. The blood then passes through into the capillary bed, labeled here, and then you see this important change in blood type occurring. So there is, recall, in capillaries only, we will have the exchange of oxygen and CO2 occurring between cells and tissues. So on the oxygen-rich side of the capillary bed, oxygen and other nutrients are going to be unloaded into cells and tissues, while on the oxygen-poor side of the capillary bed, CO2 and other waste products are going to be brought into the capillaries. So this accounts for the change in oxygen and CO2 levels that you see on each side of the capillary bed. Now, similarly, we can then transport blood out of the capillary bed through venules, which eventually work their way up into larger veins, and then eventually one of the two vena cava that lead in directly into the right heart. Now, another important structure that I have labeled here are called precapillary sphincters. These precapillary sphincters represent single smooth muscle cells that have the capability of constricting and limiting or reducing the blood flow to certain areas of the capillary bed and therefore the organ or tissue. And we rely on these precapillary sphincters to control or reroute the blood flow depending on the body's physiological needs. There are three major types of capillaries in the cardiovascular system. These are classified based on the size of pores that are found in the endothelial lining of the blood vessel wall. Starting with continuous capillaries, these are the most predominant type of capillary in the human body. These have the smallest pore size and are used for basic exchange of oxygen and CO2 between cells and tissues in most organ beds. Next, we have fenestrated capillaries, which have an intermediate pore size, and these pores are referred to as fenestra, which is Latin for window. These are slightly larger and accommodate passage of larger solutes like glucose, urea, and other substances that are larger than oxygen and CO2. These are present in organs like the kidneys, which allow for exchange of solutes and the retention of specific solutes in the blood versus the urine. Fenestrated capillaries are also located in the choroid plexus of the brain ventricle system and the ciliary body of the vascular tunic of the eye. The final type of capillary, called sinusoids, consists of the largest pore size of any type of capillary. These pores are called intercellular clefts, and these are so large that they allow passage of entire cells through the endothelial lining. These can be found in organs like the bone marrow, the spleen, and the liver, and these are necessary in these types of organs to allow the removal and breakdown of red blood cells, particularly in the spleen and liver, and also the production of new red blood cells in the bone marrow. In this diagram, you can see a specific type of fenestrated capillary inside of the kidneys, called the glomerular capillaries. These glomerular capillaries are necessary for the filtration of all of our body's blood. In fact, this performs the overall functioning of the kidney organ, which is to filter blood and remove liquid waste from our bloodstream. It is in these glomerular capillaries that larger substances like glucose, urea, and other substances that can be filtered out of blood will leave the glomerular capillaries and enter the lumen of the Bowman's capsule that is indicated here. It is at this point where fluids that leave the cardiovascular system become technically a part of our urinary system. Similarly, in this diagram, you can see a representation of sinusoids which are found inside of bone marrow, which are present in all of our bones. We rely on sinusoids which are present in a specific type of bone marrow called red bone marrow to allow blood cells, including red blood cells, white blood cells, and platelets, to enter the cardiovascular system. If it were not for our sinusoids and their large intercellular clefts, we would not have a means of bringing new blood cells into our cardiovascular system. And finally, recall that one of the major questions we attempted to address in this lesson was how is blood returned to the heart in the absence of appreciable blood pressure? We already identified one of these mechanisms in the form of valves, which are only present in veins and ensure that the flow of blood does not go back in the same direction in which it already came. Another additional mechanism to ensure that blood is ultimately returned to the heart is what is called the skeletal muscle pump. This relies on the contraction and relaxation of skeletal muscles, predominantly in the lower extremities, like in the calf muscles that you see here, to gradually milk blood further and further up veins in our lower extremities to the thoracic cavity. Once the blood returns to the thoracic cavity, we rely on a third mechanism called the respiratory pump, which relies on the inhalation and exhalation activities of the lungs to help milk blood directly to the top of the heart, where it can eventually return through either the inferior or superior vena cava.